Hi, I'm Alice and I'm a fourth year natural sciences student here at the University of Exeter. I'm going to tell you a little bit more about the group project which you undertake in your third year of the course. I think this is one of the most exciting parts about the course here at Exeter because you get to work with external clients and tackle real-world problems and produce real-world outputs. I worked in my group with a company called Labster. Now, Labster are a multinational company with offices in Copenhagen and Bali that's revolutionising the way science is being taught all around the world. They're an online learning platform and they create virtual simulated laboratories so students can deal with multi-million-pound equipment and undertake dangerous experiments, but all safely in virtual reality. I worked as a scientific content writer and my role was to decide the storyline the simulation would be based around, all of the quiz questions and all of the theory based around this confocal microscopy. Now, most scientific content writers are professors or really high academics, and all we were was just a bunch of students. But we had to work with the company just like any professor or high academic and produce exactly what they wanted us to produce, which was a simulation. One of the biggest tasks of this was to create something called the lab flow. Now, this is essentially a script of the entire simulation. It details all of the interactions that you have with your virtual lab assistant, your virtual iPad, Farmer Sam, for example, who we've included in our simulation. Just imagine trying to write a script for a play. You have to include all of the stage directions, all of the language, and it's very, very difficult. We produced this, and this is one of the main parts of the group project. To do this, we base it around a storyline on plant funguses. We picked the plant fungus powdery mildew, which is a fungus that is rife in Europe. It reduces crop yields and it's a real problem for farmers. And we wanted to teach students about this along with the concept of confocal microscopy. Not many of you will have come across confocal microscopy. Not many people are actually trained to use one. But as part of this project, as undergraduates, we were trained to use this microscope. Confocal microscopy works a lot different to normal light microscope methods. So rather than lighting up the entire sample, it scans a laser across the surface of the sample. Any light then coming back from that sample, anything that's out of focus is blocked out. So you only get really sharp, focused light. So that allows you to burrow into the sample up and down so you can get a 3D image, in this case, of the plant fungus and the leaf. We created this storyline and spoke to lots and lots of different artists. And these people are not scientists. So we had to develop our science communication skills to describe to them exactly all of the science that we wanted them to animate and put into our simulation. Now, Labster were really impressed with us and they really enjoyed working with us, so much so that they have published our simulation and it's available to play and buy online on their website. We also got invited to Copenhagen and we are now student ambassadors for Labster. So we have set up a university with Labster in a collaboration with students. And it's been really rewarding to know that we have developed something that is helping other people to learn.